Hello, everyone, and welcome to today's webinar titled Discovery of a Gene Microbiome, Interaction in Lung Cancer, Lessons Learned and Future Directions. This webinar is part of the Microbiology Virtual Week virtual event. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, click on that Ask a Question box, which is located on the far left of your screen. As a reminder, this presentation is educational and offers the continuing education credits. Click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Leigh Greathouse, Assistant Professor at Baylor University. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Dr. Greathouse, you may now begin your presentation. Welcome.
Thank you to the organizing committee for giving me the honor to be part of this microbiome keynote speaker. So as was mentioned today, I'm going to be talking about some of my research and actually what's more important, I think, today is some of the lessons learned from this research and then, of course, future directions. So as disclosure, since this is a microbiology conference, I am not a microbiologist or bioinformatician or immunologist. But what I did start out life as is a dietitian, and then got my master's in sports nutrition. And about halfway through my master's degree, I was actually diagnosed with a really rare form of cancer with stage 4. And during this time, I decided that cancer prevention was the way for me. And I decided to go ahead and get a Ph.D. in cancer biology. And during my postdoc, right at the beginning, I got really enamored with the microbiome and its ability to act as a risk marker for cancer prevention and potentially mechanisms for cancer therapy. And so through this first foray into the microbiome, and all of this training now, I call myself an expert poop collector. So all of this training has led to my current love, which is the Cancer Crusader, with my fellow Cancer Crusader team members. And I couldn't do any of this without them. So with that, I'm going to start talking about my outline for what we're going to be talking about today. I'll be first talking about the driving question behind the research that I do, give you a little bit of background between the microbiome and cancer, and then tell you the story about how this first interest in the lung microbiome began in the discovery of the gene-microbiome interaction, then talk to you about some of the in vitro and in silico studies that we're doing in collaboration, and some of the in vitro studies that we're doing in my lab, and then end off by talking about the lessons learned from doing these experiments and these types of studies, and then some of the future directions. So let's get right into it. So the driving question behind most of my research exclusively is, what is the contribution of the microbiome to cancer risk and treatment response? So as a molecular epidemiologist, I really like to look at things from model perspectives. So in this model, what is the population attributable factor of diet to cancer risk? It's pretty small. Genes contribute some to this, maybe about 10% depending on type of cancer. Obesity contributes a significant part of this, which is also related to diet. But we don't understand is how much does the microbiome contribute to cancer risk and treatment response? So this is the majority of the questions that we're going to be trying to answer today and be walking through today. But let's first start out talking about what is the relationship between the microbiome and cancer, because there's some pretty important distinctions that I want to point out. So we know that there's a lot of environmental associations with the microbiome. Two that I already mentioned, diet and obesity. Also, the smoking is associated with this. Where you grew up, where you live, geography is really critical to the composition of your microbiome. We also know, I'm sure a lot of speakers have already been talking about this, is the contribution of the microbiome to our immune system. And even your genetic makeup contributes to their composition, as well as the other side, which is the microbiome contributing to mutations and carcinogenesis, which will be the majority of my focus today. So one of the things I want to point out is this understanding between cancer initiation and cancer promotion. So we've known for decades now, most of the microbes that are actually cancer initiators, sufficient for initiation of cancer without too many other environmental factors, which is accounting for about 15 to 20% of all cancers worldwide are infectious agents. We know these to be HPV, BV, and then of course H. pylori. But what we know less about actually is what are the cancer promoters? And that's what some of my research and the research I've been doing with my collaborators are investigating. So how does the microbiome promote carcinogenesis? So bacteria can promote carcinogenesis in a multitude of ways, and we're still scratching the surface of our understanding about this. So bacteria are pathogens. They can inject, or some of the bacteria are pathogens, they can inject toxins or virulence factors that activate pro-growth pathways and inhibit the immune surveillance. Bacteria can take advantage of the disrupted barrier function and induce inflammation. And then when you have this disrupted barrier function, maybe you also have changes in the microbial composition, what some people call dysbiosis. This can cause immune dysregulation that affects cancer growth, but also can affect response to immunotherapy. And this has been demonstrated fairly decisively from several people's work, including Jennifer Wargo, Giorgio Turchiani, and others, showing that response to immunotherapy is really dictated by the composition of your microbiome before you start treatment. And I'll just note here today as well, some of the things that we're going to be looking at from my research is activation of the toll-like receptors through NF-kappa B and some of those signaling pathways. So one of the other things that I talked about was diet. So diet, as we know, is one of the strongest drivers of microbiome composition and metabolic function. And I love this slide because it illustrates kind of this impact that diet can have on the microbiome in relationship to this homeostasis versus this immune dysregulation function. So as the slide depicts, we have really two important functions noted here, or maybe I should say three important functions. First is this this mucosal barrier that has two components: the outside, which are composed of a thin mucosal layer, which houses the hopefully mostly commensal pathogens, and then the thicker mucosal layer that protects the epithelial cells from direct contact with the microbes. Now, what's really important about the diet here is that by eating what we call a healthier diet, high in fiber, other phytonutrients, that can support the growth of bacteria that degrade some of these what we call microbiome accessible carbohydrates. So they ferment them and they can produce short chain fatty acids, one of the most important ones being butyrate, which is what the colonocytes use for energy. This also shores up the barrier functions and keeps those tight junctions held together nicely, so you don't have leakage of bacterial products. And when these are really nice and tight and the mucosal barrier function is working properly, you have blockade of this inflammatory response, and you have more of an anti-inflammatory signaling. However, when the diet is disrupted, then you have a switch from some of more of these fiber degrading bacteria, namely Bacteroides. Then you have some of these mucin or glycogen or glycan degrading bacteria like Akkermansia that will then degrade the mucus and allow disruption of the barrier function, leakage of these factors, and then chronic inflammation to occur. And as I said previously, chronic inflammation is a strong risk factor for several different types of cancer, including colon cancer. So in the healthy state, what we currently understand in the colon is that there's a high diversity, which is really critical for allowing competition and stability, pathogen clearance, and immunoregulation. And within the healthy host, again, you have this great barrier function, a balance between pro and inflammatory responses. However, what we're starting to understand now, with a lot of great work from several people that look at fusobacteria and other bacteria like Bacteroides fragilis, what you start to see is this decreased diversity in microbiome imbalance, a change in metabolism, more bacteria that are decorated with factors that allow immune evasion or virulence factors, and then a diseased host that might have DNA damage, which causes changes in the microbiome function and composition. And then more pronounced is the immune dysfunction that comes along with this. Now, in lung cancer, it's similar, but there's some distinct differences, and we know a lot less about lung cancer, but I'll talk more about this in just a few minutes. As opposed to colon cancer, with lung cancer, you really have a low microbial diversity, but you also have the added benefit of this mucociliary escalator pumping out or clearing out most of the bacterial pathogens and a healthy immunoregulation and good, just similar to the colon, healthy barrier function and this good immune response or balance between the immune response. But then you have an imbalance in this homeostasis in the lung, where we're seeing is an increase in diversity because you've lost this barrier function, you've lost this mucociliary escalator, trying to clear out. You see this in COPD, you see it in cystic fibrosis, and more now we're starting to see it in lung cancer. And then within the diseased host, again, you're seeing this barrier dysfunction along with DNA damage. And that's what I'm going to talk to you a lot about today, is some of that work. So before we knew all this information, when I was starting out as a postdoc and trying to figure out if I even wanted to look at the lung cancer microbiome, I first started looking at what was the evidence to show that there was even, a reason to look at the microbiome in lung cancer. So some of the evidence that we started to find in these early stages was that smoking reduced epithelial barrier dysfunction, which we know smoking is a very strong risk factor for lung cancer. We noticed that in epidemiological studies that increased use of antibiotics was associated with increased risk in lung cancer. And as I mentioned earlier, COPD, also a risk factor for lung cancer, increased bacterial colonization. And also in germ-free mice, we also see that lung cancer development was reduced, and those that were inoculated with pneumonia. And also one of these factors that comes along with bacteria, lipopolysaccharide or LPS, can increase inflammation and lung tumorigenesis as well in a mouse model. Now, one of the interesting things that I discovered during this time was a really great paper that came out in about 2012, where they specifically deleted P53, the tumor suppressor gene, and challenged with AOM to induce colon cancer or colitis. And what they noticed in the mouse model that had P53 knocked out in the intestinal epithelial cells is that you had systemic identification of LPS. You also saw, apart from P53's normal DNA damage pathway function, you saw a separate function that was important in barrier function. So as this slide points out in the bottom, in the P53 animals that had P53 deleted, you have loss of barrier dysfunction and infiltration of bacteria. And interestingly enough, when they treated with antibiotics, you see that there's this decrease in signaling of NF-kappa B activity, indicating that the microbiome was a component of this, driving this activation of inflammation. So I want you to put a pin in this because I'll talk about it and later in some of the later slides. So when I first started into this, I was looking to understand even how to do microbiome analysis. So I had to teach myself those from scratch. But thankfully, Julie Segre became one of my co-mentors, and we walked through how to do this microbiome analysis with our current understanding, which was still pretty infantile at that time. So most people were using stool or saliva at the time. Not many people were actually using tissue from cancer samples, and for good reason. It's a very low very low biomass type of sample. I'd say at this point, I didn't know what I didn't know. But I had a lot of great people on my team, and part of Julie Segre's help in that was helping us get trained in microbiome analysis and providing us a really good bioinformatics bioinformatics tool called Pathoscope, which we use to do quite a bit of our analysis. But I have a paper out that talks all about these types of analysis if you want to read that. So I want to walk through the study design for this lung cancer microbiome study because it's critically important, and I'll talk about the lessons that we learned from this later on, and we'll dive a little bit more deeply into this. So the whole reason for doing this was because a clinical need for lung cancer treatment at the time was early stage diagnosis. And so we were looking for early stage biomarkers. The intent was to use machine learning with all of these microbial samples to identify microbial-based biomarkers that might be good as a clinical tool for diagnosing early stage lung cancer. So the two populations that we chose, which is discovery cohort and a validation cohort, because that's what you need to do to have a proper clinical biomarker. So in the lab I was in, Kurt Harris's lab at the time, they had a 30-year cohort of a case control study of lung cancer. We call it the NCMD case control study. And from this, I had access to almost 400 frozen tumor sections, both tumor and non-tumor adjacent. And over 100 of these were paired from the same individual. We also I gained access to over 40 tissues from immediate autopsy. That was a companion study with this case control study, as well as over 16 frozen lung tissue samples from biopsy, people who were going in to determine if they had lung cancer. Also critically important, I used 2 different kinds of negative controls. Now, because these tissues came from our repository and I didn't have knowledge of how exactly they were collected or could go back retrospectively and collect negative controls, I was able to find out what steps were taken from the OR to pathology and figure out what swabs I needed to take to control for contamination. We also did no template water controls and extraction from DNA extraction reagents. We also used positive controls, also critically important, from a bacteria community from the Broad Institute. So the methods that we use, also important, 2 different types of methods, sequencing, 16S and PacBio sequencing for more defined, higher resolution of species identification. P53 targeted sequencing, we did FISH to confirm the species. And we also attempted bacterial isolation for strain identification. Now, our validation cohort that we decided to use is the Cancer Genome Atlas, which has gotten a lot of press lately from some of the studies that have come out, and I'll talk about those a little bit later. Now, this was a great and not great place to go to use a validation cohort. The TCGA data is great and for looking at gene expression and survival and mutations, but the RNA-seq and whole genome sequencing data was not intentionally meant to look for microbial sequencing, specifically the RNA-seq data, which is poly-A-tailed, in which most bacteria don't carry poly-A-tails, so you're significantly biasing what microbes or that you're going to be able to identify. So that's why I say we only use it as a validation cohort, because the sheer number of tissues that were there. And it also had P53 sequence, which you'll see will become important later. So like I said, our initial thought was to look for a biomarker, clinical biomarker, using machine learning. We identified some biomarkers, but we couldn't successfully validate those. When we took a step back and we looked and asked the question, well, can we identify differences between the different types of lung cancer, adenocarcinoma and squamous cell carcinoma, to me are the most dominant types of lung cancer that you see. So when we did this analysis using hierarchical clustering and machine learning, we identified several bacteria that could distinguish squamous cell from adenocarcinoma. And what I'll direct your attention to is the smoking status at the top. You'll notice that the smoking status in black is significantly higher for squamous cell carcinoma, which is one of the drivers and risk factors for squamous cell as compared to adenocarcinoma. Some of the bacteria that we found that was significantly enriched in squamous cell carcinoma included acidovorax and Klebsiella. So then we took that information and asked, can we identify that in our validation cohort, the TCGA study? And because we had a little bit more access to finer level resolution, if we could identify what some of those actual species were. And so we identified acidovorax as well and Klebsiella both as having significantly more enriched in the squamous cell carcinoma versus adenocarcinoma. And we also were able to control for several confounders in this relationship, including lung location, emphysema, age, and sex, and antibiotic exposure. So as I kept emphasizing, smoking is a really strong risk factor for lung cancer. And so I asked if it is truly playing a part of lung cancer, lung carcinogenesis, and squamous cell cancer, we should find these if we try to classify using linear discriminant analysis between ever smokers and never smokers. And the strongest one that came out from this classifier was acidovorax. And then we next asked ourselves, well, if it's truly a response to smoking, we should see an increase in former or current smokers. And that's what we see when we look at the acidovorax abundance compared to never smokers. Then we next ask, okay, so smoking is really a key driver or a key risk factor in squamous cell carcinoma as opposed to adenocarcinoma. Therefore, we should see an increase in acidovorax if it's truly related or truly associated. And that's what we see in both current smokers and former smokers. And what I'm not showing, there's no association, no difference in abundance in acidovorax in people who are never smokers. Again, lending to this idea that smoking is part of the thing driving the abundance or accumulation of acidovorax in these lung tumor tissues. That's what we're showing here. And then I did one step further. We looked at we developed probes for acidovorax at the genus level and looked at our tissues, our tumor tissues, and looked to identify this using fluorescent in situ hybridization. And this is what I'm showing you here. And then a couple of my colleagues in the lab that were more expert in doing this type of analysis also identified the number of % positive cells, which you're seeing in these couple of illustrative patients, between 10 and 15% of acidovorax positive cells. So not only did we identify it at the gene sequencing level, we identified it also using specific probes. And the other thing that we identified too is the species of bacteria that we found was acidovorax temperans using PacBio sequencing, which I'm not showing here. And the last thing I will tell you about is this idea of P53 creating a dysfunctional epithelial barrier. Knowing that squamous cell carcinoma has one of the highest mutation loads of P53, anywhere between 70 and 80%, the hypothesis was that we should identify more bacteria of acidovorax abundance in tumors with P53 mutations. And as you can see here, when we look at the TCGA data, that's what we see. We see people with tumors with P53 mutations as opposed to wild type have stronger or more abundance of acidovorax in these tumors, indicating that loss of barrier function through mutation of P53 is allowing accumulation of acidovorax in these tumors. But we know that association is not causation. At this point, we've just identified a potential either passenger, but we don't know if it's actually a true promoter. And so that's where my colleagues come in, and they have been doing some of the research to promote this understanding. And I'll just take a step back here and say that at this point, we still have a couple of other studies that support this finding of acidovorax from Tyler Jack's lab and from a separate cohort in China that show acidovorax is identified in lung cancer from either TCGA or in a separate cohort. So we're at this point now where we understand that there's this microbe-gene association in lung cancer. And we know that acidovorax has been found in tobacco products. We know that microaspiration can bring in these products into the lung. And now we need to understand, is this just an association or does it play a functional role in lung cancer? And so with the help of my colleagues, Kurt Harris and Dr. Zhang, they've been doing a lot of work doing germ-free animal models, looking at whether or not Acidovorax temperans can actually modulate the immune system and promote lung tumorigenesis. And so they're using a mouse model, autochthonous mouse model of adenocarcinoma with mutations in KRAS and gain-of-function mutant in P53 using this CRE-LOX system. And so the design of this study was they introduced intranasally adenocre, and then every 2 weeks, they would intranasally introduce 10 to the 9 CFUs, either Acidovorax temperans or L-gasseri, which is a commensal or sham, and then do MRI at 13 weeks. And what they were able to demonstrate on the left panel is a significant increase in lung volume with the Acidovorax as compared to sham or L-gasseri. And we see that then illustrated as well in the right-hand panel with a significant increase in tumor volume from Acidovorax temperans. And what they also recently discovered was when they tested for the markers for CD8 T cell exhaustion, PD1 and TIM3, they saw in the Acidovorax temperans a significant increase in exhausted CD8 T cells, which will become important in the next few slides. So one of the other colleagues, postdocs in Kurt's lab, has also been following up with this information, looking at the relationship between pro-inflammatory cytokines and Acidovorax or Klebsiella, which we identified in these previous studies. So what you're seeing here is a heat map with the purple on the left being the squamous cell expression samples, expression data from the TCGA, and the blue being pro-inflammatory cytokines. And what you'll see is that there's a strong relationship, mainly in the squamous cell, for IL-8. And so when he looked at this and did correlation analysis looking at IL-8, we see a pretty strong signal, mainly in the squamous cell with both Klebsiella and a little bit more strong with Acidovorax, potentially indicating that Acidovorax may be driving this inflammatory signature. He went on to ask the question, can Acidovorax or Klebsiella alter T cell exhaustion? So using this really interesting 82 gene set expression signature of T cell exhaustion from this previous study, Gao et al. In Nature Medicine in 2018 from lung cancer, he applied it to these same samples, looking at adeno and squamous cell carcinoma, asking, is there a difference in this T cell exhaustion signature? And he did find a really significant difference between non-tumor and tumor paired samples in squamous cell carcinoma for this exhausted T cell signature. Then went on to look and see if you looked at Acidovorax and Klebsiella specifically, is it correlated with this CD8 T cell exhaustion signature? And if you look over to the left-hand side of this heat map, specifically the purple squamous cell tissue analysis, the gene expression is much stronger in the subset of genes. And then when you look at either adenocarcinoma or squamous cell carcinoma, you see this kind of a weak or moderate association, indicating that this Acidovorax and potentially Klebsiella may be driving this inflammatory response and be really driving that CD8 T cell exhaustion by constantly presenting the factors to the CD8 T cells and not allowing them to be there to actually regulate the balance of proliferation in those cancer cells. So with these data, we have been able to now expand our understanding of the influence of specific taxa towards differences in carcinogenesis of adenocarcinoma and squamous cell carcinoma, and potentially identifying a specific genus, Acidovorax temperans, that's associated with P53 mutations and that barrier dysfunction. And now we've shown a functional relationship with Acidovorax temperans and actually being a promoter, not necessarily an initiator like I talked about, but potentially a promoter of lung tumorigenesis, especially in people who are smokers or former smokers. And finally, with the help of my collaborators, we were showing this association with the pro-inflammatory cytokines and squamous cell carcinoma, and potentially that it's actually the Acidovorax and Klebsiella that are potentially actually driving this exhaustion in CD8 T cells and giving rise to lung carcinogenesis specific in people with squamous cell carcinoma. So I'm going to take a little bit of a switch now and talk a little bit more about the research that we do in my lab related to this idea, and more drilling down on what is the microbe-host mechanism promoting inflammation. So when I was a postdoc in his lab, I was really enamored with this idea of this difference in inflammatory response between commensals and between pathogens. So there's some strange little blips on my slide. I'm not sure where those are coming from. But so the model system that we use in my lab is Bacteroides fragilis for many different reasons. One of which is it comes in two flavors, the commensal form and the pathogenic form, which offers a really great model system to study. It's also strongly associated with colon carcinogenesis, as Cindy Sears' lab has shown extensively, especially in people with La-Famine disease. One of the things that became really interesting to me during this time was the function of these little vesicles that bacteria secrete called outer membrane vesicles, and their ability to modulate the inflammatory response. So the inflammatory response, one of the first lines of defense, is the innate immune response. And so in epithelial cells and immune cells, you have toll-like receptors that can recognize all different flavors of bacteria products. You know, LPS, flagellin, and then what we're really interested in actually is the TLR receptors that are capable of RNA sensing, including TLR7, 8, and 3. So depending on which TLRs are activated, you can get a pro-inflammatory response through NF-kappa B or an anti-inflammatory response through IFN gamma. And what are these little outer membrane vesicles anyway? They're essentially just little, the bacteria pinches off these little vesicles from their outer membrane, and they can contain various cargo, including toxins like the Bacteroides fragilis toxin and RNA. We've also, I'll show that in just a few minutes. So the hypothesis that I'm testing here is based on the fact that we know that RNA viruses can activate or engage TLRs to induce inflammation. And we also know that in tumors that they secrete exosomes that carry small RNAs or microRNAs that signal in a paracrine fashion and can activate these toll-like receptors, specifically TLR7. So my hypothesis is that it seems like bacteria are probably doing the same thing, secreting these little vesicles that probably carry small RNAs that are activating inflammation either through the toll-like receptors or maybe some other RNA sensing pathway. And so that's the hypothesis that we're working through right now is outer membrane vesicles are likely carrying these small RNAs that can regulate or modulate the toll-like receptor. And some early proof of principle that we first started to look through is the TCGA data asking whether or not is the TLR expression even different in colon cancer. Interestingly enough, colon cancer specifically, it's really significantly reduced in TLR expression, predominantly TLR3 and TLR7, indicating that this might be a mechanism of immune evasion that these cells in colon cancer are using to allow themselves to expand and survive. Some of our proof of principle initial experiments that we've been doing, we've been using TLR reporter cells. This is the experiments we've done so far looking at TLR4 reporter cells, and we were exposing them to OMVs for about 24 hours using the positive control of LPS for activation. And you see a difference between two different amounts of eTBF OMVs or NTBF OMVs, with the ones from eTBF activating TLR4 as opposed to eTBF, which goes along with some of the previous evidence published by other folks. And we were doing some preliminary investigations, doing co-culture experiments with OMVs and either colon cancer cells or breast cancer cells, and tracking these, trying to figure out where exactly are these OMBs going and their small RNAs, and is it different between eTBF and NTBF? And so one of the other proof of principles that we were able to look at is differential loading of these small RNAs in between NTBF and eTBF, the pathogen. And so I was able to do this experiment with the help of my collaborator, Garth Ehrlich, and his also collaborator, Josh Mell. And we set up this experiment where we collected RNAs, small RNAs from the whole cell from both strains, as well as small RNAs from their outer membrane vesicles. And we applied RNA-seq. And so Josh Mell was instrumental in helping us do the bioinformatic analysis of looking at and aligning all of these small RNA reads to either the NTBF or the eTBF reference genomes. And this is pretty much showing just RNA genes only. And what we see is actually a really big difference in what small RNAs are expressed in the whole cell on the right-hand side of the heat map, as opposed to what the small RNAs that are expressed in the OMBs. And most interesting to me is that there's this little brick of genes that small RNAs that are actually overexpressed specifically in eTBF outer membrane vesicles, lending credence to this idea that small RNAs may be specifically engaging nucleic acid receptor, toll-like receptors, or RNA sensing pathways in the cells. And so that's some of the preliminary work that we've been doing, and we're following this up and replicating it with outer membrane vesicles from Acidovorax temperans using either a triculture model where we have polarized KCO2 cells in combination with Raji-B and goblet cells, or in my lab in collonoids as well. Also following up looking at the co-localization and subcellular localization, and then doing some RNA protein analysis, looking at what RNAs are actually associated with these TLRs using his clip and RNA-seq. And this is supported by two of my really close colleagues, Joe Tobby and Christy Saves, that we're doing this work together with. So that culminates a lot of the information I'm going to talk to you about with regards to the lung cancer microbiome story. And now I'm going to do a really hard switch and talk about the lessons learned. And this, I think, is actually even more critical than my research, is understanding how to do this type of research appropriately, because a lot of people are really interested in doing microbiome research, especially human microbiome research, and digging in their freezer, pulling out those samples, and not knowing exactly how to set this up. You can get some really bad microbiome data. So I'm going to walk you through how to do this. So why should you care? Well, DNA extraction actually really contributes the most of variation to studies. A lot of studies fail to report sample collection procedures, DNA extraction, and one of my pet peeves is background contamination control. So all of this leads to a reproducibility failure and lots of false leads that we don't need or want, especially in the clinic. We want sound, valid, reproducible biomarkers that we can use in the clinic and follow up with for functional analysis. So my colleagues and I, Rashmi Sinha and Emily Voteman at the NCI, wrote this paper detailing out, especially for low biomass studies, which I'll describe, what are the key points you need to keep in mind when you're doing these human microbiome studies. So study design is first. So how are you collecting? Especially if it's a low biomass study, it needs to be sterile collection techniques, which I'll talk about in a second. How are they being transported and stored? That's a really big, problem right now, is if you're collecting samples and they're being transported, what's happening to them in transport? Or, what is the type of storage material that you're using, buffer or whatnot? Also, are you carefully understanding how you're extracting them? Are they two different people extracting them? Did you check your reagents for microbial contaminants? So all of this I'm saying because in two different groups, from the Microbiome Quality Control Group and the International Human Microbiome Standards Group, from both Europe and the United States, all diverged or all converged and said the majority of experimental variability from these human microbiome studies was from DNA extraction. So that's why I'm talking about this so passionately. So low biomass samples are mainly what we think of as less than about 10,000 microbial cells. And what we were trying to get across in this paper was that you really need to go beyond just sequencing and show what we call proof of life. So you need to use culture-based methods to try to culture out bacteria from these tissue sections, hopefully fresh tissue that you have, as well as following up with fish or fluorescent in situ hybridization for those specific bacteria that you're looking for. And then potentially, something like qRT-PCR as well. So let me give you some examples of low biomass samples that you should proceed with caution for sure. Fresh frozen tissue samples, paraffin-embedded tissues, bronchial lavage tissues, skin swabs, environmental samples, anything that has low biomass, you really need to do your due diligence and understanding what exactly each step was in collecting, storing, and extracting that DNA. Because as the example shows here, you can have 2 different samples with 2 different treatments. And depending on how that DNA was extracted, maybe it was 2 different people, maybe it was different lots of reagents, you could get contamination DNA so much that it actually shows a signal that actually isn't biologically relevant. And you have completely missed the boat on something that could have been really important, but because you didn't control for contamination, you're not seeing it. So let me give you some examples of how to proceed with caution. So recently there was this really important paper that came out with the first author portal in Nature, and they did similar to what I did. They did a much more extensive profile of all of the TCGA data. Now, I told you there's inherent bias in the TCGA data because the way it's collected, you don't know what the contaminants were, but they also used blood samples from this as a means to control, as well as developing a lot of other controls kind of in silico to potentially get rid of some of those contaminants, and then built predictors based on this. And the important thing is they validated this signature, or attempted to validate this signature with circulating tumor DNA from blood samples from a completely different cohort, which is really critically important to validate. And so they were able to, in a subset, validate some of these signatures in cancer. And so the next study that came out most recently from the Nijman et al. First author in Science was a similar attempt, but instead of using frozen tissue, they did use a few, though. Their main objective was looking for bacteria that might be intracellular in all of these different types of tissues. So they looked at brain, lung, pancreas, skin, uterus, breast, and bone. And they all used almost exclusively paraffin-embedded tissues. Now, if you can imagine how paraffin-embedded tissues are created and what types of microbes might track with those, from OR, so you have, and these are historical samples, these aren't created, most of them weren't created new. So you have in the OR, you don't know what all of the bacteria are going to be collected on the tumor tissue itself from the ink that's being used, the different tools that are being used to grow the tissue. They're not sterile unless you create an aseptic procedure, which you can. So then you have the question, well, what microbes came in the paraffin bath? Are there microbes that are lingering in there? So they did do some extraction of just the paraffin to control for contamination. They also used some specific probes to look for bacteria. And importantly, they also did do some specific fresh tissue bacteria colonization to identify viable bacteria that are actually found from the sequencing. So they did do some of that more extensive validation procedures like I talked about, but we still need to do a lot more validation and better job of doing collecting samples from the start. So an example of best practices in collecting these samples is setting up a sterile pathology suite. And this is one of my colleagues from Mayo Clinic who set this up for collecting microbiome samples from surgery. So you need to know what are all the steps from surgery to pathology, where the tissues could collect freeloader passengers, microbes that shouldn't be there. So you can set this up even in very small spaces, and you just need to do your due diligence and go there and figure out where all the places that tissue touches, and can I sterilize it before, or do I need to collect samples of it later on? So another really good paper that illustrated some ideas to combat this contamination issue for low biomass studies was the RIDE principle from this paper in 2019. So report experimental design, include controls like we talked about, determine the level of contamination, and explore contaminant taxa with each study and interpreting the biological results. So really important. So in the last part of my talk, I'll tell you a little bit about some of the future directions we're taking with my research. So really looking mainly at colon cancer and the microbiome response. So remember I told you my driving question was, what is the contribution of the microbiome to cancer risk and treatment response? But what is really important that a lot of studies miss is, what is the contribution of diet and the microbiome to response to treatment? Because as I'll point out in just a few minutes, diet contributes a significant component to the microbiome composition. As illustrated in one of my favorite studies, which was this 2-week diet exchange between rural Africans, which have some of the lowest incidence of colon cancer in the world, and African Americans. And within just a 2-week diet exchange, with the main component difference being fiber, they saw massive changes in biomarkers of cancer risk, butyrate increases, and changes in the microbiome composition. So indicating with just 2 weeks significant dietary change, you have significant changes in the microbiome. Sorry, I think my slides got a little messed up here. And another illustration of this, how important diet is to the microbiome in cancer response, from Jennifer Wargo's group at a past meeting, they demonstrated that diet was actually really important in response to immunotherapy. In fact, they found that people that had been treated with immunotherapy, they found that patients who had consumed a high fiber diet were about 5 times more likely to respond preferably to the anti-PD-1 treatment compared to those on a low fiber diet, indicating that fiber is a significant contributor to response to cancer therapy. So this was part of the rationale in designing this study, to look at the interaction between diet and the microbiome and how it influences cancer therapy response and toxicity. One of the main concerns that we see with colon cancer patients is this huge number of people dealing with chemotherapy-induced diarrhea. It's really problematic, especially in people undergoing FOLFOX treatment. And up to 5% of those actually die from this chemotherapy-induced diarrhea. And unfortunately, there's no clinical biomarkers for it. The current strategies are insufficient at this point. And it also has all the hallmarks of mucosal damage and inflammation, indicating that there's a microbial component to this. And in fact, there was a study done several years ago showing that there was a significant change in the microbiome over time as somebody was exposed to radiotherapy in the microbiome in association with diarrhea. So this prompted me to put the study together to address whether or not the diet or the microbiome or the combination thereof was actually predictive of chemotherapy-induced diarrhea. And hopefully maybe we can use that information to do something to treat these people, or even to prevent CID, either through the diet or changes in the microbiome. And so we're going to be recruiting through DoD funding recently this study to recruit people with stage 2 or 3 colon cancer, about 120 individuals. We're going to start by setting up a sterile pathology suite to collect those tissues and blood and fecal samples, as well as food frequency questionnaires at baseline. And then before treatment, we'll get another dietary information, stool and blood, and then multiple times throughout chemotherapy, we're going to be collecting stool, blood, and dietary information and doing follow-up up to 5 years. And so hopefully we'll be able to provide some maybe biomarkers or information for diet to prevent or reduce this burden of chemotherapy-induced diarrhea in colon cancer patients. So the ultimate goal of this research, I hope in the future, is actually to identify what are these tipping points in the microbiome that promote carcinogenesis. So if we understand what those tipping points are and find them before carcinogenesis gets a hold in that tissue, then perhaps we can prevent carcinogenesis occurring overall, or improve prognosis and treatment response. And so understanding what these tipping points are is a key component of my lab. So I'd just like to acknowledge all of the folks in my lab, my 2 doctoral students, Julia and Dill, and my new doctoral student, Mahir, who I didn't have a chance to put on here yet. And then several of my undergraduates who are listed there. The Tabi lab has been really instrumental in a lot of our in vitro studies with outer membrane vesicles. Garth Ehrlich's lab, instrumental again with the Bacteroides fragilis studies. And then my previous lab, who I still collaborate with extensively, and Kurt Harris, and looking at following up on those lung cancer microbiome studies. My biostatistician at Mayo Clinic, June Chen, James White, my bioinformatician, and of course my funding resources there. If you want to contact me, I'm at Cancer Microbes, especially if you are looking for a postdoc position. We are hiring a postdoc. If you have skills in bioinformatics, microbiology, or microbiome analysis, please get in touch with me. My Twitter handle is down there, as well as my email. So I am happy to take any questions.
Thank you so much, Dr. Greathouse, for that informative presentation. And we will now start the Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in from our audience. Dr. Greathouse, are there any foods which may increase the risk of especially lung cancer?
Well, I would say that that's a really great question. Most of these foods which increase cancer risk in general have been mainly focused on inflammatory related cancers. Lung cancer is one of them, but mainly colon cancer. So without saying specific foods to reduce lung cancer, I would say foods that are anti-inflammatory that generally reduce your inflammatory profile. So eating lots of fruits and vegetables and whole grains. In fact, I was just giving this lecture in my nutrition class. Anything that reduces your overall inflammatory milieu in your system. So making sure you get plenty of physical activity so you're not obese. Making sure really it's just eating a lot of colorful fruits and vegetables with whole grains as the majority of your diet. I like the 80-20 rule for myself. It works out well. 80% of the time I eat as healthy as I can, whole grains. You don't have to eat organic. You don't need to do that. But, you know, whole grains, whole fruits and vegetables, cooking for yourself, and 20% of the time you can eat whatever you want. And usually that gets you to a point where you have a good balance of pro versus anti-inflammatory milieu in your body.
Thank you for that. Our next question is, what sort of effect does one's epigenetic have on microbiome composition and chances for lung cancer?
Yeah, that's a really great question. I think that's some of the things that we really don't understand yet. And that's a question that I have myself, is we just don't understand very well how does the microbiome either change or interact with your epigenome. We do know that butyrate, some of the short chain fatty acid, can act at different concentrations as an HDAC inhibitor. So understanding it has a difference when you're talking about lung carcinogenesis versus how it acts when you already have cancer, or colon or lung cancer overall. So that's a great question. We still don't really understand what that connection is between epigenetics and the microbiome. But it's definitely an open for research in that area for sure.
Thank you so much. And I want to thank our audience members for these great questions coming in. Alongside a diet, what else can one do to help change their microbiome composition and decrease the chances of having lung cancer?
So one thing is don't smoke, and especially don't vape. That's terrible. So those are the two main things that you can reduce your risk of lung cancer. Physical activity. If one of the other things besides diet that changes the microbiome really drastically is physical activity. If you could see the difference in the cluster analysis when you look at somebody who's highly physical active versus sedentary, the difference is so dramatic. So I can't speak to enough about increasing your physical activity and reducing your risk for lung cancer, because when you train your lungs, you're exercising them and preventing any of those microbes that might be pathogenic from hanging on, because you're constantly expiring those microbes out, those pathogens out of your lungs by doing that physical activity. And when you're breathing in all of those other microbes, it's training your immune system to be responsive to all of these other pathogens. So when we think about our pandemic, COVID-19, and who is being more likely to get sick or die from these, it's people who have obesity, people who are elderly, people whose immune system is really poor or not as helpful as somebody who's young, who's physically active. So I think that would be my answer to that question.
Thank you for that. And I think that's actually a really important point right now, as many people are sitting behind the computer all day long. What diet or eating style is the most optimal for the microbiome? And you mentioned fiber consumption. Are there any other diet specifics that you would recommend?
Well, I can answer that really easy. The Mediterranean diet. Mediterranean diet is probably one of the best diets that you can eat for a very healthy microbiome. It's high in whole grains. It's high in anti-inflammatory fruits and vegetables. The main component of Mediterranean diet is really healthy, brightly colored vegetables along with whole grains, and healthy fats like olive oil, not saturated fats like that you find in animal meats or red meats. And with regards to fiber consumption, if you don't think you can get enough fiber in your diet, there's other prebiotic, formulations out there like Metamucil. There's a new one on the market called Prebiotin, and they dissolve in your water or your coffee. And you can drink this to increase your fiber intake and help, improve your microbiome composition. It also, sometimes it helps ameliorate some other problems, digestive issues that you've got going on. So Mediterranean diet, absolute best diet.
Thank you for that. We have time for a couple more questions. Are probiotics and prebiotics effective in preventative care for colon cancer?
There are some preliminary evidence out there showing that mainly prebiotics could be using as a preventative for colon cancer. Probiotics, I am not a fan of right now because we just don't have enough information about these different strains that they're using. And when I tell people about probiotics, if you are a healthy, normal individual with, probably no GI issues, you do not need a daily probiotic. That is just silly. And you're wasting your money. It's like when you take multivitamins, you're giving yourself expensive urine. If you take probiotics, you're just giving yourself expensive poop. So you need, for a probiotic, you only need these in special circumstances, especially, the really high potent ones. If you have an ongoing GI disorder, there's a new one out for IBS called Holy Goss or Holygos is a new one that's being on the market for IBS. For specific situations, there are probiotics, but for a daily general use, you do not need to take them.
Wow, that's good information. Thank you so much. We have time for one more question. Oh, actually, we have a couple more coming in. Let me go ahead and give you these last two. How can one introduce microbiome into lung cancer cell lines that are in vitro studies?
So in vitro studies, you're probably talking about the anaerobic nature of a lot of these microbes. So there are certain ways that you can set up your cell lines in an anaerobic chamber. Some of these anaerobes, depending on what kind they are, will last for about 24 hours in cell culture in norm oxia conditions. But you can also set up organoid cultures. So my colleagues at Baylor College of Medicine, Rob Britton, and his former postdoc, Jennifer Octoon, who's now, I think, at Utah or Ohio, actually set up these conditions where you can actually culture anaerobic bacteria with normal or cancerous monoculture cell lines. And they've got that system working out pretty well. And in my studies, what I'm actually using is just the outer membrane vesicles. So we don't have to worry about, oxygenation at that point. We can just, take out the outer membrane vesicles and then expose them to the cells. But there's several people, Cindy Sears' lab, Rob Britton's lab, Jennifer Octoon have worked out these properties to co-culture anaerobes with standard cell lines.
Thank you so much. Our last question, is there a huge difference in the microbiome content between people who are genetically in risk for lung cancer and people who have developed them due to lifestyle or environmental influences?
That's a really great question. I don't think we have the data quite yet to be able to address that. That would be a really great study to set up. You'd have to collect a lot of background information on and have huge numbers to really dive into that question. But yeah, it'd be a really interesting question. I mean, the only thing that we know right now is, smoking, which is the risk factor for cancer. But any of the other genetic markers for lung cancer, we don't have a whole lot of information on those differences for lung cancer between those people. So, but it's a great question.
Dr. Greathouse, this has been a great presentation. Thank you so much. Would you like to provide any closing remarks for our audience before we go today?
Yeah, just, again, we're open for postdoc position. Please feel free to contact me if you are interested, or if you know somebody that's interested, or you have colleagues that have students that might be interested in that position. So thanks again.
And thank you again. You've inspired me to go out for a walk for sure today.
Great, I'm glad.
And I thank all the audience for their participation and joining us and their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 6 months up until March 2021, and LabRoots will alert you via email when it's ready for replay. We encourage you to share this email with your colleagues who may have missed this live event. Until next time, be safe and stay healthy. Take care. Bye-bye.